Hello friends, myself Sripad Bairagi from MES College of Pharmacy, Sonai. Today we are going to learn about the neurohumoral transmission. Firstly we will start with the definitions of the neurohumoral transmissions. The transmissions of the neurons, transmissions of the signals across the neuron by releasing the different types of the neurotransmission. Previously the junctional transmissions was carried out by using the different sorts of your electrical impulses. But the Dell's scientists and some of them like scientists proven that the release of the chemicals are responsible for the interconnect between the transmissions of the one neuron to the another neurons. That is also called as a vagus stuff. The Dell scientists perfuse the two frog hearts in a series and stimulate the hearts by the vagus nerve, and by the releasing of the transmitter it shows that there is an arrest of both the hearts, and that's why this vagus stuff is known as the transmissions of your particular neurotransmission. Let's start with the properties of the neurotransmitters. Neurotransmitters are nothing but the chemicals. Previously it is known as the electrical signals, but now the chemicals also responsible to interfere with the one neuron to the another neuron and stimulate the second neuron also. So different properties are there that each and every neurotransmitter should have. Only then we can say that these transmitters are effective and process the transmissions of the signal from one neuron to the another neuron. The first property is that it should be released presynaptically, then only the communications will be effective. It should be released upon the stimulation. Unless and until receive the electrical stimuli or particular stimulation, it should not be released. That is the second property of the transmitters. Third one, its application show the particular types of the response. It may be responsible to induce the effect or to decrease the effect. And the last one is that the effect which is produced by the transmitter, it should be the antagonized with the similar antagonist released by the particular transmission. So these are some few properties of the neurotransmitter it should have. Then only we can say that the transmission will be effective. Now we'll move toward the steps involved in the neurotransmission. The neurotransmissions are releases. Prior to that, the release of the neurotransmitter, the synthesis is the very important step. And definitely, whatever the biogenic amines are there or which act as a neurotransmitter that are synthesized inside the body within the specific area, that is called as a synthesis of your particular transmitter. After the synthesis of the transmitter, these transmitters are stored into the synaptic vesicles. So the first step we consider, that is after the synthesis of the transmitter, that is the impulse conduction. Impulse conduction, we know that the impulse are very important for the transmissions of the signals. So impulse conduction in which the RMP, that is the resting membrane potential, the difference in the ionic composition intracellularly and extracellularly, which is responsible or which can be recorded as a voltage. So the resting membrane potential is always a negative, which indicates that there is a high concentration of the negative ions intracellularly as compared to the extracellularly. So that the negative potential or resting membrane potential that maintain within the minus 70 to minus 90 millivolt. So upon the stimulation or the arrival of the electrical impulse, there is an increasing of the sodium conductions, which results into the opening of the faster opener sodium channels, which open fastly and also close fastly. That results the entry of the sodium channel intracellularly, and which causes increasing the membrane potential near about the plus 20 millivolts. As soon as the sodium enter inside, the potassium ions start to leaving the cells and to maintain the ionic composition effectively. So this is the first step in which the impulse conductions takes place. Now the second step, that is the release of the transmitter. The transmitter, which we can call as, depending on the nature, that is either they are the excitatory in nature or the inhibitory in nature, which are stored synaptically or at the axonal terminal inside the vesicles at a prejunctional nerve ending. As soon as the action potential arrives from your axon to the axon terminal, there is a fluidization of the membrane causes by following the entry of the calcium intracellularly. As soon as the calcium enter inside, whatever the secretory vesicles that came into that are fused with the axon terminal, and by the process of the exocytosis, there is a release of that your neurotransmitters takes place. Now come to that the third step, that is a transmitter action on the post-junctional membrane. So depend on the nature of the neurotransmitters. The neurotransmitters are combined with the specific types of the receptor. We know that each and every transmitter have the specific types of the receptors. Then only this transmitter can interfere with that or interact with the receptor and show the actions. So depending on the nature of the transmitter, it shows that the action, that is either EPFCs are there or IPFCs. That is nothing but the excitatory postsynaptic potential or inhibitory postsynaptic potential. Excitatory postsynaptic potential, in which we can take an example by releasing of the positive or the positive neurotransmitter. When it interact with its receptor, it causes that the increasing the entry of the specifically the cations. Likewise, the sodium is there or the calcium is there, and that causes that the increasing the depolarization. In short, we can say that it responsible to increases the activity. And IPFCs are nothing but we can call as the inhibitory postsynaptic potential. When the inhibitory neurotransmitter to bind with its own receptor, it causes that the opening of the particularly the cation, for example the chloride ions, which causes the decreasing the depolarization, nothing but the increasing the hyperpolarization, and shows that the relaxation like effect. Then the last point, that is the post-junctional activity. Post-junctional activity is nothing but when, depending on the nature of the neurotransmitter, when it binds to that particular receptor, it shows that the EPFCs or the IPFCs. If the EPFCs are there, it is responsible for the generate a propagated post-junctional action potential, which results into that the increasing that the neuro or the nerve impulses and leads to that the increasing the contraction. Whereas IPFCs, nothing but which stabilize the post-junctional membrane and causes that the resistance to depolarization stimuli. It means that it inhibits that the depolarization state and causes that the increase that the hyperpolarization. And the terminal or terminations of the transmitter actions. Transmitters that are maybe depending on the acting, the locally acted, isn't it, or they act the other sites, isn't it? So we can say that they act on the specific receptor or the another receptor. For example, transmitter, the termination of the transmitter is also that important step. If the neurotransmitters are act a longer time to particular receptor, it may causes that the paralysis of that particular site. So it is necessary to terminate the transmitter. So the termination of the transmitter can be takes place by the locally, or whatever the released transmitter that is taken back into the circulation. So that we can call as it's a reuptake mechanism will be there. Some few examples are explained here. Likewise, the terminations are transmitter that is takes place by the locally termination. For example, the acetylcholine itself and terminated by the actions of the acetylcholine esterase. Second one is that by the actions of the specifically reuptake mechanism. Likewise, the NET, that is a specific carrier proteins or norepinephrine transport protein, by which the concentrations of that your epinephrine from the effector site will be decreases. So in this way, the terminations of the transmitters takes place, and the axonal membrane is again ready to receive that the next stimuli, and the action can be shown. Friends, in this diagram we can see all these actions via that, particularly by looking to the each and every numbers, starting with that the first one, that is the motor neuron action potential. So similarly that we discussed the generations of the action potential or conductions of the action potential, as the action potential is a travel from that dendrites to axon and toward the axonal membrane. So here you can see that the action potentials are traveling from that particular sites to that this axon terminal. As soon as the electrical impulses or the activity reach to that your axon terminal, there is a release of the transmitters takes place. So that's why here they are putting the second point, that is the entry of the calcium through that the voltage gated ions, and by which as soon as the calcium enter inside, you can see that these vesicles are fused with that the axonal membrane, and after the fusion by the process of the exocytosis process, there is a release of the acetylcholines takes place from this axonal membrane. Then whatever the release acetylcholine, as a neurotransmitter, it have its own receptor. So this acetylcholine can bind to its own receptor. Here you can see that the acetylcholine receptors are there. Once the acetylcholine bind to its receptor, it shows that the different kind of the actions. For example, you can see that it leads to that the positive actions. So the sodium enter inside, and as soon as the sodium enter inside, the local current within that the postsynaptic membrane that is increased, and leads to that the sodium causes the depolarizations of the end plate potential, and the contraction is facilitated. That it. So here you can see that the voltage gated calcium channels are sodium channels are open along with that the calcium. So the muscle fiber action potential that is initiated. As soon as the muscle fiber action potentials are initiated, which results into the propagations of the action potential from one area to the another area. So in this way the transmissions are begin, and at last here you can see that the acetylcholine degradation. In last point we already discussed, that is the terminations of the neurotransmitter. So this release excess quanta, we can say that whatever the excess quanta of the acetylcholine that is degraded by that your acetylcholine or the enzyme choline esterase. Here you can see that the choline esterase enzyme, which is responsible for the hydrolysis of the acetylcholine into acetate and choline, thereby the net concentration of the acetylcholine at the axonal term ending that is decreases. So friends, in this way actually the neurohumoral transmissions is begin. Thank you friends.